about 7,500 different enzymes or proteins, and you have about 8,000 chemicals. This is, for example, for plants, for humans, for pyramid of life. This is for animals. The same pyramid, the metabolome is connected to all other arms. So you see, and the metabolome or metabolome is connected to the proteome and the genome. There is an interconnection between the different arms. And this is the environment influence or how the environment, and this is what makes metabolomics more complex. Because you can see you have 25,000 genes, and at the end you have 8,000 metabolites. Which means probably there is many genes they can encode for the same metabolite, because you see you have 8,000 metabolites, you have a 7,500 enzymes, so it's a similar number. In another way, you have to understand which genes that encode, that control the biosynthesis of that metabolite. So this is, it just describes the brief, history of the omics technology. So from the gene, then you have the transcript, or the transcript, the RNA, and then it goes to the protein. So you see for one gene, it encodes for different RNAs. Then you have different proteins, and you have specific metabolites. And from analyzing the metabolomics, so you have the store, compare, and cluster, integrate. So this is, we will discuss, we will develop that, later. So, and then you have the biological knowledge, raw food improvement, genetic and metabolic engineering. So the result or the outcome of that, so you can manipulate or you can, design the different plants, because you understand not only the genes, but you understand the metabolism of that plant, of that gene. So this is an example. The gene, the target material, so you have the genes, chromosome, genetic code. In the case, so what happens, the molecular weight, about 100,000 to about 120. The contexts are independent because the genes, they are independent. For few exception, all the genes are independent. Mapping and sequencing, we sequence the genes. As we see, we know the sequence of the genes. And the DNA sequence. The DNA sequencer. By DNA sequencer now, you have automatic sequencing. You just purify the DNA and you put it in the sequencer, and after a few hours you get the sequence. And even now you don't need to sequence all the gene. You just sequence a portion of that gene. Then after we use what we call bioinformatics, and by bioinformatics you can determine the sequence of the gene. The transcript, so it's the code. So you have the same number. So you see approximately the same number. Then you have the context dependent, because some RNAs, they can be dependent. Sequencing and the protocol, we do what we call the hybridization. You know the process of hybridization? I think you did genetics. Not all of you, but The hybridization is the fact to make a new portion by using a probe. So I do not want to go to the process of, this is molecular biology. So you can just read about the process of the protocol of hybridization. For the proteins, so you see from that huge number of genes, you get about 5 to 20,000 proteins, with about less, maximum 20%. So if you estimate a ratio, you have about at least 5 genes that encode for one protein. If you have a 5,000, it's means 20 genes, they can encode for one protein. A minimum of 20 genes encoding for one protein. So between 5 to 20 genes, they can encode for one protein. So you see what the complex. So which gene, because you have one protein, so that means at least 5 up to 20 genes they can encode for that protein. And that's what makes proteomics more complex than the gene, because the gene, as we said, what we are interested in the gene, in the omics, because all the omics, our goal is what is the expression of that gene. What we are interested in the result, how this gene expresses phenotype or a metabolite. Example, you know that the fruit, you take a fruit, you go to the market, you buy a fruit, you put it on the counter, and after a few days, you have it browning, and there is a decay, and it spoils, right? The freshness is, so what happens? Of course, there is some genes, but the result you are interested in, that, for example, browning and of flavor, so there is some metabolites. So, and probably there is many genes that encoded for that, for example, ethylene, because we know that ethylene, accelerates the senescence of the fruits. And finally, so you see, you have a 5,000, which means by 1,000